Good day everyone. We are going to discuss about the dark matter of biology. So what is the dark matter of biology? Dark matter inside cells remains mostly hidden and is likely to have multiple vital roles to regulate signaling, rates of reactions, water structure and viscosity, crowding, and other cellular activities. So dark matter biology is interacting and performing functions that are perceptible, yet we cannot directly detect or sense the matter itself. So the species that we cannot or do not see belongs to the dark matter of biology. So there must be something we do not know. There must be something we miss in studying what is inside the cell. So Milo and Phillips detailed the average ionic nature inside and outside of cells.
Good day everyone. So I am Giselle B121 and I'm going to discuss to you about the viscosity and the crowding in connection with the dark matter of biology. So the viscosity and crowding. So the interior of a cell is a complex fluid, so in which is defined as a mixture with presence of microscopic length. So it plays an important role in our community and it has a different characteristics that helps us in their work. So they have different length, which is from the small molecules to the macromolecules and to complexes to organelles, just like our pyramid of life from cells, etc. So this person, Miss Catherine Luby Phelps, has written several nice articles reviewing the literature while giving a wonderful description and introduction about the interior of the cell. Miss Catherine Phelps concludes that the interior of the cell is a complex fluid. Mato yung gising, complex siya. And it is heterogeneous. So there are variety of metal ions that have specific functions in the cells. So each of them has specific roles, and first one is nearly 50% of proteins require the coordination of metal ion species to function. So dark matter of biology is just like what Hani said. It is hidden, so ngit-ngit siya. But then there are different species nga belong ato niya nga object, and just like the proteins, mo na siya ang yang role, na siya 50% nga rolling sa katong specific thing. So the calcium di ba for the bones, di ba sir? It regulates myosin motors and a number of actin-binding proteins that can regulate muscle contraction, such as troponin and tropomyosin. So there are many ions in the community, but actually the magnesium is obviously the most used and highly abundant in cells.
Hi, I'm Marshie and we're going to tackle about the intrinsically disordered proteins and domains. So almost 40% of the proteins identified in the genomics data have a stretch of 50 or more amino acids that is intrinsically disordered or unstructured.
What's up?
So 40% of the proteins in our DNA or genomics has been identified as disordered. But these disordered pairs have certain functions that helps the cell. So 15% of all proteins are completely disordered. according to function. So our proteins have definite functions in the cells, but 15% of them are disordered. So this table shows the percentage of peptides. This table shows the percentage of peptides with unstructured region of specified length. So as we can observe, almost 80% or 70% of the proteins are unstructured or disordered. So next is functions of the intrinsically disordered proteins. So first one, they act as polymers. Next is act as entropic springs and molecular spacers. Entropic springs and molecular spacers aid purposely regulating the distance between structural colloidal regions of the objects or different pairs of proteins. Next is termed by Peter Tompa as fuzzy states. Fuzzy means coiled or disordered proteins are multiple conformations or change in shapes in proteins. It is characterized as random coil states. Next are examples of disordered proteins. First is the tau protein, important in axon development and maintenance. Tau protein have a definite role in the cells, but most of the ordering and structure are actually bad things for tau and cause it to aggregate into neurofibrillary tangles that are associated with frontotemporal dementia and Alzheimer's disease. So, these tau protein are one of the reasons why a person suffers from Alzheimer's disease or frontotemporal dementia. Next is the tubulin dimer. Its carboxy terminal is disordered but highly charged. So, not necessarily the tubulin dimer is the whole protein that is being disordered, but only its carboxy terminal. The role of its carboxy terminal is essential to microtubule form and function. So without the tubulin dimer carboxyl tail, the microtubule cannot perform its specific functions in the cell.
Go.
Next is the post-translational modifications. It is a process involving signaling like phosphorylations in the cell. Its downstream effect can alter network formation, rigidity, stability, and carbon transport. Mostly occurs in the filaments of the cell.
Action.
Good day everyone, I'm Elaine and I'm going to discuss about the weak and transient interaction. So weak and transient interaction.
Go.
Weak and transient interactions are easy to miss or ignore because they are difficult to detect. Instead, most initial studies focus on are easy to miss or ignore because they are difficult to detect. Instead, most initial studies focus on strongly interacting species that have long binding items. Because weak or transient interactions would be difficult to determine in dilute biochemical assays. Another is, weak and transient interaction are almost impossible to detect with traditional biochemical assays. This is because conventional biochemical experiments rely on strong interaction that can withstand long preparation times and dilute conditions. So, in this journal, it says they use the microscopy imaging with single molecule sensitivity to visualize weak, transient, fluorescent species as to interact. Then, they predict that weak interactions are not only common but immensely important in the cell. Such weak interaction can locally increase the concentration of important molecules in a specific region.
So the dark matter biology, like biophysics, is to assume that many things don't matter, or many things don't matter rather.
Focus! Yet, time and time again, we are foiled by biology. Because it appears that many more matter or many more things matter than we assume. and some things that we think matter do not matter to the process we are focusing on.
I'd like to acknowledge Ms. Jennifer L. Rose for her journal entitled The Dark Matter of Biology. Thank you, ma'am.
Sir, thank you.
Good.